What is a prokaryotic replication fork? Well, let's start with what are prokaryotes. Prokaryotes are unicellular organisms that are broken down into two different domains of bacteria and archaea. For this video, we'll be looking at bacterial cells and how they replicate their own DNA. A bacterial cell contains circular DNA that programs all of its cell functions. On the bacterial chromosome, there is a specific sequence of DNA that acts as the origin of replication. The origin of replication is where DNA replication starts in the bacterial cell. The replication can proceed in both directions of the DNA, and this is called bidirectional replication. Prokaryotes have one origin of replication on each bacterial chromosome, while eukaryotes have multiple origins. DNA replication begins when an initiator protein binds to the origin of replication on the chromosome and causes the unwinding of the double-stranded DNA. Multiple proteins are needed for this unwinding process. From the origin of replication, as the DNA segment unwinds, the section of DNA that is undergoing replication and unwinding is called the replication bubble. The points that mark where the double-stranded DNA separates into single strands on either side of the replication bubble are called the replication forks. Let's discuss the process of unwinding and the important proteins that are involved. Helicase is an enzyme that breaks the hydrogen bonds between the nucleotides, which unwind the DNA double helix. There are also single-stranded binding proteins bound to the single strands of DNA to maintain strand separation, protect the DNA, and avoid environmental interactions that can cause secondary structure formation. Outside of the replication fork, there is the enzyme topoisomerase, or more specifically, gyrase, which relieves the supercoiling or tension that forms due to unwinding of the DNA by nicking the DNA. DNA is antiparallel, meaning one strand runs from 5' to 3' and another runs 3 to 5'. 5' and 3' indicate the carbon numbers along the sugar backbone in DNA, where the 5' carbon has a phosphate group attached and the 3' carbon has a hydroxyl group attached. This is important because the enzyme DNA polymerase III needs to build from the 3' end and is responsible for adding nucleotides to the new DNA strand. RNA primase is a type of RNA polymerase that adds RNA primers to provide a 3' hydroxyl group end for the DNA polymerase III to build off of. At the replication fork, there is a leading strand and a lagging strand. The leading strand runs 5' to 3' in the direction of replication and is formed from continuous replication after an RNA primer is attached at the origin. The lagging strand runs in the opposite direction and is discontinuous replication, where RNA primers are placed along the strand forming DNA fragments called Okazaki fragments. As replication is occurring, DNA polymerase I removes RNA primers and replaces them with DNA. As DNA polymerase III and I add nucleotides to the new DNA, they catalyze phosphodiester bond formation, which is the bond between adjacent nucleotides. After DNA polymerase I removes RNA primers, ligase is an enzyme that seals the break in the sugar phosphate backbone between Okazaki fragments by catalyzing phosphodiester formation. As the prokaryotic replication forks replicate the entire bacterial DNA structure, the forks cross and divide the new and old DNA to complete the DNA replication process.